Okay, so 41 is animal nutrition. Okay, so there are for animals there's a lot of essential things that they need to live. It's kind of like plants. Yes. No. Anyways, there's essential amino acids which include isoleucine, leucine, lysine, whatever. All of these and also valine and babies, they're kind of different. They need histidine. Anyways, so these essential nutrients are substances that like animals have to have, but they can't just like really make them, so they have to get them from somewhere else. And you can get these amino acids and proteins from like a lot of different sources, which includes animal sources and plant sources, but the protein in animals and plants are a little different. So in proteins, the meat and like meat and eggs, these proteins are complete proteins, but plant proteins are incomplete. So I guess like having animal protein is better than plant protein. And also animals don't have the enzymes to form double bonds in certain fatty acids that they need to like live. So fatty acids, a lot of them have to be obtained through food. So through linoleic acid, that is the essential fatty acid. Okay, here are a list of vitamins that you guys, I mean that humans need. So there's like many different types of vitamin Bs. There's vitamin C, which is ascorbic acid, which is like it's basically citric acid. You can get it in sour fruits and stuff. There's also vitamin A, which is retinol. It's good for your eyes. Um, Also vitamin D, which is good for your bones. You can get that in dairy, vitamin E and K. And also these are mineral requirements of humans. So they need humans will need calcium, phosphorus, sulfur, potassium, chlorine, sodium, and magnesium in like really large quantities because those will make up a lot of your um body compounds. And then mineral there's also other minerals such as iron, fluorine, and iodine, which you'll need in smaller concentrations. So iron is really important for your red blood cells for the for hemoglobin formation. That's why you need iron. And if you don't have enough, you have an iron deficiency, you can't get enough oxygen, then you'll like have anemia, which is like lack of oxygen. Yeah. Okay. Okay, so these are your steps of food processing, which is basically when you turn food into your energy and then your waste. So there are a lot of different feeding mechanisms that organisms will use. This includes filter feeding, substrate feeding, fluid feeding, and then bulk feeding. So filter feeding is used by a lot of aquatic animals where basically they will strain smaller organisms or food particles from the water or the medium around it. So an example of that would be like certain whales, I guess they filter feed little shrimps or fish or something. Okay, and then substrate feeding is when you have an animal that will live on its food, like a caterpillar, it's gonna live on, yeah, krill. It's gonna live on the leaf that it's eating. Fluid feeding is when you have an animal that sucks nutrients in the form of a fluid from its host, like a mosquito will suck your blood. That's fluid feeding. And then bulk feeding is when you're eating a big piece of food at one time. So like humans. And basically all big animals will do that. Okay. So in food processing, there is 4 smaller processes. The first process is ingestion, which is the actual act of eating or feeding. And then digestion is where the food is broken down into smaller molecules for the body to absorb. And this is done through breaking bonds through hydrolysis, which is the adding of water. And then absorption is when these animal cells will take up and absorb the nutrients. And then elimination is when undigested material will pass out of the digestive system and be eliminated. Okay. So these are the different types of digestive compartments that animals will use to digest their food. There are a lot of different types because we're all different. So certain organisms will use intracellular digestion, which they will basically have a food vacuole, like a little pouch in which the digestion will, oh wait, no, never mind. That is food vacuoles. So that is when a cell will basically use its cell membrane to take in food through food vacuoles through endocytosis, and then digest that in the vacuoles with their lysozymes, and use hydrolytic enzymes to break down their food. So endocytosis, I think we've talked about it before, that includes phagocytosis and pinocytosis. So pinocytosis is taking in really small molecules, and phagocytosis is taking in bigger molecules. So extracellular digestion is the other type of digestion where it's digested like with not inside of the cell, basically. So the first type of extracellular digestion involves a gastrovascular cavity, which is part of a simple body plan. So that gastrovascular cavity, it has one opening, and it's kind of like a pouch. So that pouch is responsible for digestion and also like distributing nutrients around the body. And then undigested particles will be released through the same mouth hole again. And then in complex body plans will have a complete digestive tract and an alimentary canal, which includes a mouth and an anus. So it has two holes, and it's like a big tube in the middle. So in this type of digestion, the food will move in one direction only, like it's gonna pass from the mouth to the anus. And then that has the alimentary canal will have a lot of different specialized parts for like ingestion, digestion, absorption, and excretion. So there will be a lot of different organs for that. And then it's compartmentalized, which also allows for ingestion of new food when the old food is still being digested. So in the gastrovascular cavity, when you have old food, you can't take in new food without it being mixed, but then in these alimentary canals, you can. Okay. So your food processing will begin in your mouth, which is your oral cavity. So when you are anticipating food, like when you're about to eat food or when you are eating food, that will trigger the release of saliva by salivary glands. So that will help like moisturize your mouth cavity, because saliva will contain mucus that lubricates the food for better swallowing. And it's going to protect your gums against abrasion. And also saliva will facilitate your taste and your smell. So other parts of saliva also include certain buffers that will prevent tooth decay by food, by neutralizing acid, and like it also has some antimicrobial agents to like kill any bad bacteria. And your saliva will also contain a thing called amylase, which breaks down starch and glycogen. And this will help release food particles stuck to the teeth and also reduce nutrients that are available to microbes in your mouth. So amylase is kind of the reason why, like, if you have like a cookie or something, you hold it in your, like, not even cookie, just like rice, and if you hold it in your mouth for a long time, it's gonna start tasting kind of sweet. Yeah, amylase will contribute to that. And then anyways, you're gonna like chew the food with your teeth, and your tongue's gonna shape your saliva and your mushed up food into a bolus, and that's gonna get pushed to the back of the oral activity and then into the pharynx, which is the throat region. So there's a thing called an epiglottis, which when you're breathing, it's open to allow the air in, but when you're swallowing, that's gonna close and to like prevent the food from going into your lungs. So when you choke when you're eating food, that's because your epiglottis is not down and you got food or fluid into your lungs. Okay. Anyways, the pharynx, which is the throat region, it's gonna lead to the esophagus, which is the long tube that connects to the stomach, and also the trachea, which is gonna connect to your lungs. And your esophagus, it's gonna push the food along by peristalsis, which is basically alternating waves of smooth muscle contraction and relaxation. So when you eat, that's not your food going down isn't just because of gravity, it's also because of muscle contraction. So technically you can't eat upside down. Anyways, at the end of your esophagus around here, like where the stomach begins, there's a sphincter that will regulate the passage of ingested food into your stomach, so it doesn't go all at once into the stomach. It's kind of like a drawstring bag type of thing. Okay. Next is where chemical digestion happens in the stomach. So chemical digestion is done with hydrochloric acid. You might know that. Hydrochloric acid is super acidic. It has a pH around 2, which is very acidic. That's gonna disrupt your extracellular matrix in your meat and plant material and basically break those cells down, denature their proteins and expose their peptide bonds. So these exposed peptide bonds, I'm just going to go into a little bit of detail, but they are attacked by protease, which is also known as pepsin, and protease will basically, they break down proteins. That's what they're called, protease. They will break down these proteins into smaller polypeptides and then expose more contents of ingested tissues. So that's how like it generally works. So hydrochloric acid is produced by parietal and chief cells like near the stomach. These parietal cells will use an ATP pump to like expel hydrogen ions into the lumen, and then chloride ions will also diffuse into the lumen, which is like around here. In these like, I don't know what to call these. Anyways, the chloride ions and the hydrogen ions will then react and form hydrochloric acid, and yeah, they will diffuse through, and then in the lumen they will start forming hydrochloric acid to prevent these parietal cells from like being digested as well. And so and then chief cells will release the pepsin, which is also protease. It's going to release that into the lumen in the inactive form of pepsinogens. So hydrochloric acid will convert pepsinogen into active pepsin by exposing its active site, and this also only forms in the lumen to prevent the parietal and chief cells from being digested. What? My bad. Okay. Anyways. Oh. Sorry about that. Um, anyways, so to like protect the stomach against digesting itself, there's gonna be mucus that's secreted by cells, like oh. Okay, wait, I'm gonna like reshare because I don't know. Okay, I'm sorry if that happens again. I'm not really sure what's going on either. Okay. So there's gonna be mucus secreted by cells and in the glands to protect against damage by the hydrochloric acid. And there's gonna be cell division on top and like the inner layer of the stomach that adds a new epithelial layer every like around 3 days. So that's gonna also be protecting the stomach against damage. And these cells will replace the lining before it's fully eroded usually, but sometimes this doesn't happen properly, and that's going to result in gastric ulcers, which is basically damaged parts of the stomach because of the acid and pepsinogen and pepsin. So regarding stomach dynamics, the stomach will basically churn the food and it's going to churn its contents to facilitate chemical digestion by bringing all the food into contact with the gastric juices, and that results in the formation of a thing called chyme. And that chyme is and the stomach is going to move the material through the alimentary canal because there's going to be other peristaltic contractions that will move the chyme from the stomach into the small intestine. Okay, that's the digestion the digestion part. And here, if you see in the diagram, there's two sphincters, one on each end, one right before the small intestine and one right before the stomach, that regulate the flow of food. Okay, the small intestine is responsible for both digestion and absorption. So the digestion will happen in the first part of the small intestine, which is the duodenum. There's gonna be you chyme from the stomach that came from the stomach, and then that's gonna mix with digestive juices from the pancreas, liver, gallbladder, and other gland cells. So when this chyme arrives, it's gonna trigger the release of secretin, which will stimulate the pancreas to secrete bicarbonate, which is basic. So a basic thing will neutralize an acidic thing. So your bicarbonate is gonna neutralize the acidity of chyme and act as a buffer for chemical digestion in the small intestine, so it doesn't damage the small intestine much. And then it's also going to release other proteases like trypsin and chymotrypsin, which are produced initially as inactive forms and will be activated when it's in the lumen of the duodenum. And also the epithelial lining of the duodenum will secrete certain digestive enzymes that will help with the digestion of food. Also fats, when they're in the small intestine initially, they're not digested very easily because they're like usually big globules, but they will be digested by bile salts. And those are basically like detergents that break apart fat and lipid globules. So bile production will also help destruction of certain red blood cells that are not fully functional. And there will be pigments that are released, which are incorporated into the bile pigments and eliminated in the feces. And the bile is produced in the liver, but sometimes bile pigments will accumulate in the skin when there's a liver disorder and cause something called jaundice, which is when your skin becomes yellow because of these bile pigments. Okay, and after digestion, the small intestine will start absorption. So the contents of the duodenum will move by peristalsis into the jejunum and ileum. I don't think you need to know these names. They're just like the second and third parts of the small intestine. So in the small intestine, there are things called villi and microvilli, which are projections, like little projections that contain blood vessels. So villi are little, like villi will contain a lot of capillaries, and then microvilli are like even smaller projections on the villi. So these projections will increase surface area for maximum nutrient absorption. And microvilli and villi will form a thing called like a brush border with a really brushy texture. So the capillaries and veins in the villi will carry nutrient-rich blood away from the villi and converge into the hepatic portal vein, which directly leads to the liver and then travels to the heart. So the blood will first go to the liver because the liver can remove a lot of toxins before the blood enters other parts of the body and can convert nutrients into other forms. So I don't have a picture of that, but this is just like a diagram of all the enzymes that are needed through digestion and absorption and everything. Okay. Next is the large intestine, which consists of the colon, the cecum, the rectum, and the appendix. So the colon is the large tube here that will complete the recovery of water. So that will take a good long time, but when the water is recovered, the end product is called feces or poop, and that consists of undigested materials and other wastes. So the cecum here is a little pouch that is important for fertilizing substances. So that is like right by the small intestine. In a lot of herbivores, their cecum will be really long because they need a lot of time to fertilize the hard plant material that they're eating. And humans have an appendix, which is an extension of the cecum in the human that can be used to store microorganisms, but sometimes it's removed because you get an inflammation there. And then once your waste, your feces move along the colon, it's going to be stored in the rectum until they can be eliminated. Okay, so there are evolutionary adaptations for digestion of and eating and digestion of food. So one of these includes dental adaptations. So it's pretty obvious that different organisms have to have different teeth for eating different things. So in carnivores, they have relatively sharp teeth for tearing through the meat and like chewing the bones and stuff. In herbivores, they have like sharp but like flat teeth for biting plants. And then in omnivores, they have both like sharpish teeth and also flat molars for grinding food. And then there's also stomach and intestinal adaptations. So like I said before, so plant eating animals have a really long cecum here because they need a lot of space for like for the fertilization, not fertilization, oh my gosh. For the for the food to like process a little longer because they're eating very starchy, very hard plants. Yeah. Okay. And then there are also there's a microbiome, which is a collection of microorganisms and their genetic material in the gut. And that will help provide certain vitamins that we can't produce ourselves. And these have a role in our in preventing obesity, preventing nutritional deficiencies, and also preventing diabetes. So they're very important in your gut. Okay. So next is the regulation of digestion, appetite, and energy usage. So your digestion is going to be paused when there's no food intake because you don't have to expend energy on that. So peristalsis will be controlled by the enteric nervous system, which is responsible for the digestive system. So your energy storage is also determined by the amount of calories that we intake and expend. So when we intake more calories than we expend, the energy is going to be stored as glycogen or fats. And when we expend more energy than we intake energy, we're going to have to start breaking down the fats and glycogen to release energy for our cells to keep working. So oh, and also hunger can also be regulated through the hypothalamus, which is part of the endocrine system. Okay, so glucose homeostasis is pretty important because that talks about insulin and glucagon. So when your glucose levels in your body go up, you will produce more insulin for the cells to take in that glucose. So insulin will help your cells take in glucose, but when your blood sugar levels drop, your body's going to produce more glucagon to release glucose from your energy storage side, such as your glycogen. So a disorder in glucose homeostasis is diabetes, which includes type 1 or type 2 diabetes. So in type 1 diabetes, your you'll have an autoimmune disease that will destroy your insulin producing beta cells, and then your cells can't take in glucose even if there's a lot of glucose. But in type 2 diabetes, you're going to have like insulin produced, but your cells still can't take up the glucose. And obesity can be a factor that plays in type 2 diabetes. All right, next we're going to be talking about 42, which is circulation and gas exchange. Do you have any questions about the previous one? Okay, I'm gonna move on first. Okay, so the circulatory systems will help link exchange surfaces with cells throughout the body. So that's gonna kind of link your body fluids and your blood to like exchange gas, nutrients, and waste. And the general like process that dictates gas exchange is diffusion. So oxygen and carbon dioxide, which are the two main gases being exchanged during gas exchange, they will diffuse from higher concentrations to lower concentrations. That's your basis of gas exchange. Okay, so there are many different types of circulatory systems. In cnidarians, they're gonna have the gastrovascular cavity, which allows for distribution of substances through the body when it branches throughout the system, so they don't have a circulatory system. But a lot of other organisms will. So circulatory systems have two basic types. One is the open circulatory system, and the other is the closed. So in general, circulatory systems have a heart that will power circulation by consuming energy to pump the fluids around. And in an open circulatory system, your fluid is going to be hemolymph. And this hemolymph will also be the fluid that is bathing the cells. So there's nothing to completely contain the hemolymph. So the hemolymph will flow into the interconnecting spaces between the cells. And when your heart relaxes, the hemolymph will be drawn back into the heart. When it contracts, the hemolymph it's going to be like expelled into the body. And then using hemolymph takes less energy because there's less pressure to maintain, but your organisms are generally going to be smaller because you don't have anything to like guide your hemolymph around. And then in a closed circulatory system, you're going to have blood as your circulatory fluid, and your blood's going to be confined into vessels, which are different from your interstitial fluid that is bathing the cells. So the chemical exchange is going to happen between the blood and the interstitial fluid. And worms, cephalopods, and all vertebrates will have a closed circulatory system. And this allows for more active and larger animals because of the high pressure that you can generate because of the vessels, and that allows for further delivery of blood. Okay, so the cardiovascular system will contain of the heart and the blood vessels. So the general flow of blood is first from the arteries, which are away from the heart. They're gonna branch out into arterioles, which are smaller vessels that lead blood away from the heart, and then into capillaries, which are very small blood vessels that lie in capillary beds and carry out gas exchange. Then that's going to convene into venules and then to veins, which lead to the heart. So inside the heart, there are atria and ventricles. So the atria will receive the blood from the vessels, and then the ventricles will pump the blood out into the vessels. Okay, and then the last important thing here is single versus double circulation. So in single circulation, basically one single loop of blood will return from where it started. So like in fish, the blood started out in the heart, it's gonna end up directly in the heart. And in double circulation, you're gonna have one pump to oxygenate your blood from the lungs, and then another pump to to deliver the oxygen to the body, and also receive wastes from the body as well. Okay. Yeah. Hi.
Hi, so cartilaginous fish and bony fish, they're different phylums. So do all fish have like the single circulation?
Well, damn. I'm not sure about that. I will see. I will see that. Okay, I'll double check on that. Thank you for asking though. I'm gonna assume they're similar, but I'm not sure.
No, but they're different phylums entirely. They're supposed to be like they just they didn't they don't have a common ancestor.
I see. Okay, I'll check that. Okay. Okay, so this is double circulation in mammals. Your mammalian circulation is gonna follow a certain order. So like if you assume your blood is starting at the right atrium with deoxygenated blood that's returning from the body, which is here, that's the right atrium. It's gonna go from the right atrium to the right ventricle, and then out to the pulmonary artery into the lungs, where it's gonna get oxygenated, and it's gonna return to the left atrium, go to the left ventricle, and then out through the aorta, which is the big artery that leads your blood away from the heart. And then that's gonna eventually go through your arterioles into your capillaries, go into the venules, go into the veins, and that blood's gonna end up in the vena cava, which is the big vein that leads all the blood to the heart. So you have a superior vena cava and an inferior vena cava. Your superior is gonna be from your head, and your inferior is gonna be from your superior is gonna be from your upper body, and then your inferior is gonna be from your lower body. And then that's going to return to the right atrium, and the whole cycle is going to begin again. So the cardiac cycle is one complete sequence of contracting the heart and then refilling with blood. So we have certain names for that. Contraction is also called systole, and relaxation is also called diastole. So the amount of blood that's pumped out per cardiac cycle will be determined by your heart rate and your stroke volume, which is how much blood is pumped per contraction. And then inside of the heart, there's going to be valves in the heart that will prevent backflow, because mixing of oxygenated and because in general, mixing of blood can be kind of painful. So atrial ventricular valves, which are AV valves, they're between the atria and ventricle. And then there's also semilunar valves, which are at the exits of the heart to prevent all the blood from going back into the heart. So the closing of the valves will create the sounds of a heartbeat. So that's what you hear when you use a stethoscope. And then when these valves don't function properly, that creates heart murmur, which is when blood squirts through an abnormal valve, and that's not very good. Okay, so heart rhythm will be maintained by nodes, which includes the sinoatrial and the atrioventricular nodes. So the heartbeat is not controlled by the nervous system, because if it was, you gotta have a heartbeat when you're not actively thinking about it. So these nodes are basically pacemaker cells that will generate their own action potentials and signals to make the heart beat. So the SA nodes will start that process, start making the signals, and then that's going to be delayed for a little bit at the AV nodes, and then eventually these signals are going to be passed to the heart apex, which are at the ventricles, and then your heart's going to pump. So that is also why your heart is generally has thicker muscle on the bottom, because your heart pumps from your ventricles out to the top, which are where your arteries are. Okay, and then these signals can be displayed on an electrocardiogram, which is also an ECG or an EKG. They're both the same thing. Okay. Oh, okay. Okay, so here are your blood vessels. All of your blood vessels are gonna have a smooth endothelium that will line the vessel, and these are really smooth, so that's gonna reduce the resistance of blood flow. And there are 3 general types of blood vessels: capillaries, arteries, and veins. So capillaries are the smallest blood vessel. They're only about 1 blood cell in diameter. They're very small, and very thin, so that allows for better gas exchange. And then vessels and arteries are thicker with more layers. So the layer next to the endothelium consists of smooth muscle and elastic fibers that will help with the constriction and dilation of blood vessels, and also regulating the path of blood flow. And the outer layer is a layer of connective tissue that contains really flexible elastin fibers and also collagen that will strengthen the blood vessel. So arteries in general are thicker than veins, because they have to counteract the really high pressure of blood when it gets pumped, because there's nothing to slow down the blood first when it's pumped. So veins, however, they have to contain valves to prevent the backflow of blood, because you don't want blood to mix backwards, because often valves, they can be going up your legs, so they have to contain valves to prevent that. And also the pressure in veins are much less than in the arteries, because the blood flow will slow significantly in the capillaries, which reduces the blood pressure when it goes to the veins. So your blood pressure is going to change during the cardiac cycle. So the blood pressure of the arteries is the highest when the heart contracts at systole, and this is called systolic pressure. And then your blood pressure is going to be relatively lower when the heart relaxes, which is called diastolic pressure. And blood pressure is often regulated through vasoconstriction and vasodilation. Vasoconstriction is Vasoconstriction is the shrinking of blood vessels, essentially, so that's going to increase your blood pressure. And vasodilation is going to decrease your blood pressure because dilation is the expansion of the blood vessel. And there's a substance called nitric oxide, which will reduce the vasodilation. I mean, sorry, not reduce, it's going to induce vasodilation. Okay. So capillaries, they are controlled by sphincters at the start of capillaries. That's going to control the blood flow into the capillaries. And these capillaries, since they only contain the endothelium, they will not be able to regulate blood flow. So these sphincters will help like stop blood flow at times and start them again. And small molecules inside of the capillaries can diffuse across the endothelial cells or microscopic pores for like gas exchange, for example. And finally, there's going to be fluid that's left over from this process. So all of that fluid is going to be returned from the lymphatic system. So lymph is the returned fluids from the blood because sometimes you just have extra blood fluid left over. Though that's going to drain into large veins at the base of the neck, and lymph is also filtered by lymph nodes at the base of the neck that are filled with white blood cells that can fight any infection in the lymph. So that's also why when you get sick, if you feel here, your lymph nodes are located here, I mean, you will have big lymph nodes here, and that's gonna get even bigger when you're sick because the white blood cells are fighting infection there. Okay, here's the blood components. They function in exchange, transport, and defense. So plasma is the liquid connective tissue matrix that everything else your blood cells and your platelets are suspended in. Your platelets are small cell pieces that help with blood clotting, and they will activate thrombin to turn fibrinogen into fibrin fibers that help block up parts of the blood vessel. And erythrocytes are red blood cells. They don't have a nucleus. So they initially will when they're developing, but they will lose that nucleus, and they don't have that to fit through the capillaries and the capillaries, and they can contain hemoglobin to reversibly bind your oxygen. And if the hemoglobin isn't working, that's going to create sickle cell disease, which is very painful and results in erythrocytes that are not well formed. Next are leukocytes, which are white blood cells. There are 5 different types of white blood cells, and they help fight infection. Platelets, like I said, are your clotting factors, and stem cells. Your blood will also have stem cells. They can reproduce indefinitely to produce any type of blood cell. And so LDL and HDL. So that's relevant to cardiovascular disease. So cardiovascular disease is caused because of components in the blood building up, such as a lot of fatty deposits hardening and building up. So that creates atherosclerosis. In atherosclerosis, which we'll use as an example, the key developer, I guess, is cholesterol. So cholesterol in the body is good because it's gonna be in the cell membranes to keep fluidity, but if you have too much, that's not good for your body. So the body has low density lipoproteins, which are LDLs. They will return the cholesterol to the body for the cell membrane. And also high density lipoproteins, or HDLs, they will return the cholesterol to the body for the liver. So if you have a high HDL to LDL ratio, you're more likely to get atherosclerosis. And atherosclerosis is very, very bad because they can block very vital blood vessels. So people with heart attack, that's because of damage to the cardiac muscle due to blockages in the blood vessel. And people can also get strokes, which is death of brain cells. Hypertension? Oh, I forgot what hypertension was. Anyways, a stroke is the death of brain cells because of a lack of oxygen as a result of blockages and yes, okay. Yeah. As a result of blockages in blood vessels. So that is what can happen if you have buildups of components in your blood vessels. Okay, next is gas exchange. I'm on slide 20 of 50, bro. Okay. Okay, so the respiratory media is the substance that the gases enter the body through for gas exchange. This can be air for humans, for example, or water for like fish. And the respiratory surface is the part of the body that the gas exchange occurs in, like the lungs or the gills. So in gills, there's going to be a process called ventilation, which is the movement of the respiratory media over the respiratory surface, basically like the water washing over the gills. And the efficiency of the gas exchange is going to be maximized by counter current exchange, which is like a component of ventilation. That's when water will flow over the respiratory surfaces with blood in the opposite direction that the blood is flowing. And this is very efficient because at every point of contact, the blood will be less saturated with oxygen than the water, so the oxygen will diffuse into the blood more easily. And insects are a little different. They have a tracheal system that have air tubes open to the environment that allow for direct gas exchange. And organisms with lungs, lungs are like big air sacs that allow for breathing and gas exchange. So the path of airflow in is basically gas flows in through like the nasal cavity or the mouth that goes into the larynx, which is the upper part of the respiratory region near the throat. It's going to go to the windpipe, which is the trachea. It's going to move to the bronchi. There you have two bronchi, which one leads to each lung. That's going to branch out into bronchioles, and then eventually into alveoli, which are clustered air sacs where the gas exchange will occur. So these alveoli are like kind of like little compartments or like little balloons. I don't know if that analogy makes sense, but the alveoli won't collapse because of the production of certain surfactants that will coat the inside of the alveoli. And then on the alveoli, there's a lot of capillaries that aid in gas exchange, and that's where your gas exchange will occur. Okay, so breathing. There are two types of breathing. This is for lungs, by the way. There is positive pressure and negative pressure breathing. So in positive pressure breathing, such as in amphibians, they're gonna push their air into lungs, kind of like swallowing air, but like pushing the air into the lungs instead. And then in negative pressure breathing, which is what we use, we pull the air into the lungs. It's kind of like a syringe where we pull back the plunger to get the air into the thing. And this happens because we have a diaphragm, which is a muscle at the base of the lungs. So when we inhale, the diaphragm will contract to basically pull the lungs bigger. And when we have a bigger lung, we have more space for air to move in, so that air is gonna move in. Yeah. And when we exhale, the diaphragm is gonna relax, which makes the lungs smaller. So the amount of air inhaled and exhaled with each breath is called tidal volume, and that's gonna depend on your age, your gender, and occupation, and also lung condition. And so the tidal volume during your maximum inhalation and exhalation is called vital capacity. And any air that remains after forced exhalation is called residual volume. And like you can't exhale all of the air in your lungs. That's just not very possible. And don't try it, please. Okay. There are respiratory pigments that will help with the binding of oxygen, and those circulate with the blood or the hemolymph. And that includes hemoglobin, which is what we have on our red blood cells. So a very interesting thing with regards to respiratory pigments is the Bohr effect, which is when, like for example, hemoglobin's affinity for oxygen is reduced when the environment it becomes more acidic. So that's really good for our cells because carbon dioxide, it is very acidic. it's not very acidic, but it's acidic. So when there's a lot of metabolic activity, you're going to have more carbon dioxide. So in a more acidic environment, your oxygen is going to be released. And so there's going to be more oxygen for the cells to use when there's a lot of metabolic activity. And in muscles have myoglobin, which have the same function as hemoglobin, but basically stores oxygen in the muscle for later use. And a lot of diving animals have a lot of myoglobin, which allows them to dive for longer without coming up for air. Okay, I'm gonna try to finish this. We're gonna talk about the immune system, or like get started on it. Okay, so the immune system, the basics are like pathogens are not good because they're gonna steal the resources and harm the body, harm the host. So we gotta get rid of them. So there's two ways to prevent or get rid of them. So in defense, we try to keep the pathogens out. So we have skin, or other animals are gonna have exoskeleton to cover up the body to physically prevent pathogens from entering, and also mucus secretions will help trap pathogens. And then on the other hand, we have offense, which is when we attack the pathogen, when the pathogen gets into our body. So for that, we have immune cells that will recognize self from non-self with receptors on the pathogen. Okay, so here is the innate immunity of invertebrates. So innate immunity, basically, it's very general and will happen for like every pathogen. It won't adapt to a specific pathogen. So that's what happens for every pathogen that we get in our body. So sorry, this is invertebrates, but vertebrates will have the same thing. So for invertebrates, they will have chitin, which acts as a shield, or a lysozyme that breaks down bacterial cell walls. And then for cellular defense, for hemocytes, hemocytes are the immune cells of an insect. So there are phagocytic hemocytes which recognize antigens and eat them up. Secreting hemocytes, which will release antimicrobial peptides, and also toll receptors that will coordinate the release of antifungal peptides. So that is for insects. Okay, anyways, here is our innate immunity. So we also have barrier defenses like skin, mucous membranes, tears, whatever. And our tears, saliva, and mucus have lysozymes, which will help like digest pathogens that enter. And then cells will also have certain defenses. So we also have toll-like receptors, which will bind to epitopes, which are like binding sites that are specific to pathogens. We have macrophages and neutrophils, which are phagocytic, and they will like eat up the pathogens. So macrophages they're basically big eaters that will why mucus? I don't know. I didn't make the slides. Okay, macrophages are big eaters that will stay in one place or migrate to different tissues. They're really big cells. And neutrophils are smaller cells that are also phagocytic. And they will circulate in the blood and are attracted to infected tissues by chemical signals. And macrophages are obviously bigger than neutrophils. Okay, and then there are also innate defense cells, which are dendritic cells that will activate the adaptive immunity once innate immunity doesn't work. It's found in tissues in contact with the outside. And also eosinophils, which target multicellular pathogens like parasites. And also natural killer cells that will release chemicals that kill infected cells and it won't directly kill the pathogen, but it's going to kill the infected host cells. Okay, so the local inflammatory response is going to occur when foreign particles damage the skin. That's going to involve the skin becoming really warm to the touch, red, and swollen. And this is caused by the release of histamines, which are released by mast cells. And histamines will basically cause more fluid and blood flow, which increases the temperature and makes the surface look more red. So, in the local inflammatory response, so for example, if you have a splinter in your skin, your macrophage is first going to come and engulf the bacteria, which will release cytokines. So cytokines will then recruit neutrophils. Like I said before, neutrophils are attracted to infected tissues by chemical signals such as cytokines. And mast cells will then come and release histamines, which will cause the capillaries to widen, which allows more fluids, and then more neutrophils and more antimicrobial peptides. So this is going to keep happening with a positive feedback until all of the pathogens are dead. So there's going to be basically more macrophages, more cytokines, and that's just going to try to like destroy all the pathogens. And then eventually the end product of that basically is pus, which is like a whitish liquid that has a lot of white blood cells, dead pathogens, and damaged tissue debris, and then that's going to be taken up by the lymphatic system and like filtered out.
Wait,
So
I have a question.
yeah,
So, like, in this picture, it's like after the splinter's removed, right? Like
so
all the left.
initially the splinters in there. After that, yeah, the splinter's already removed. Yeah. Okay. And then a more severe injury or infection can lead to a systemic inflammatory response, which is like throughout the whole body. Okay. So a systemic inflammatory response is due to like a severe injury which releases a lot of neutrophils at once. So it can result in a fever to increase your body temperature and possibly eliminate pathogens, there's but also to possibly increase the effectiveness of phagocytosis or also speed up chemical reactions so that the body can repair faster. But then a really, really huge systemic inflammatory response can result in septic shock, which is really bad because that can result in super high fever, really low pressure, and poor blood flow. And some people can die of septic shock. And this is common in elderly and infants. So yeah. Okay. And okay. So then here are antimicrobial peptides and proteins. So interferons are proteins that will like interfere with viral replication. It's going to prevent the viruses from replicating in the first place. And those interferons are going to make other cells also inhibit viral replication. So the complement system is where you have a lot of proteins that can basically bind to epitopes and like dig a hole in them to cause fluid inflow into the cell and or like fluid inflow into the pathogen and cause it to lyse and explode. Okay. So in adaptive immunity, receptors will provide pathogen-specific recognition, and it's good to know that adaptive immunity can only happen in vertebrates. Okay, so here is an overview. So adaptive immunity will utilize B and T cells, which are lymphocytes, and both are produced in the bone marrow. B cells will mature in the bone marrow, which is why they're called B cells, and T cells will mature in the thymus, which is why they're called T cells. So an antigen is anything that elicits a B or T cell response, and it's specific to every pathogen. And they're usually really large proteins or polysaccharides. So okay, so B and T cells will have antigen receptors that will bind to an antigen, and they're going to bind on the epitope, which is a really small and specific part of the antigen. So it's not going to bind to the entire antigen. And each antigen can have a lot of different epitopes. So they're basically the binding sites. Okay. Okay, and I'm just going to keep going. B cells, they will have antigen receptors that have 2 light chains and 2 heavy chains. So your light chains are the shorter ones, your heavy chains are the bigger ones. Each of these chains will have a constant region that remains basically the same, and also variable regions that have more variation in the amino acids, and the variable regions are on the ends because that's where your antigen binding sites are. So your B cells are activated by first binding to an antigen. It's usually by like random matching, so whichever one binds first will match with the whichever one matches with the antigen will bind with it first. So after binding, the B cell will proliferate and then make more clones that can make antibodies, which are also known as immunoglobulins. And antibodies are basically copies of the antigen receptor that are soluble in the blood and will circulate in humors. And then T cells, they will also have antigen receptors that are kind of different from B cell receptors. So they will have an alpha and a beta chain instead of a light and heavy chain. So the antigen receptors will only bind to fragments of antigens that are displayed on host cells through major histocompatibility or complex or MHC molecules. So certain host cells, they will like take in the pathogen and then eventually express the antigen that the pathogen had. Yeah, so like the host cell is taking in the pathogen and expressing the antigen here. Okay. I'm gonna do like one more slide. Okay, so when a new antigen appears, the cells are gonna have to be activated. So when there's a new antigen, B cells will lymphocytes will be randomly matched to it. And then when the match is made, the lymphocyte's going to proliferate into identical cells that have the antigen receptor. And then these cells will either form effector cells, which are short-lived and will act right away against pathogens with the same antigen, or memory cells, which are more long-lived, will like roam around your body and can give rise to effector cells if the same antigen is present in the body again. And this is why vaccines can work relatively long-term, because your memory cells are just going to be stored in your body. And this is also called clonal selection because the antigen will basically select which lymphocyte will clone based on the match. Okay, and this immunological memory from the memory cells, this is B cell activation, actually. This immunological memory will be responsible for long-term protection against a pathogen, like I said. So initially, the response is going to be called the primary response, which happens with the first encounter with the pathogen. So that response is going to be relatively slower because the body has to match up the B cell with the antigen and then let the cells proliferate and then make effector cells or memory cells. But then in the secondary response, the response is going to be a lot faster and more intense because the body already has some memory cells that can quickly become effector cells when the antigen is presented again. Okay, I'm going to go over time because we started late. Okay. So adaptive immunity will defend against the infection of body fluids and cells. So the helper T cell will basically start this whole process. So the helper T cell can activate humoral and cell-mediated responses. Humoral responses are basically cells attacking pathogens, and cell-mediated responses are like killing infected cells. There are two conditions for that. First, the helper T cell has to bind to the specific antigen, and also the antigen has to be presented on an antigen-presenting cell. That can be a B cell, a macrophage, or a dendritic cell. So antigen-presenting cells are different from normal cells that can display antigens. So like I said before, any cell basically can display antigens as long as it takes in the pathogen and brings up the antigen on an MHC complex. But normal cells only have class 1 MHC, but antigen presenting cells have both class 1 and class 2 MHC. So when the helper T cell is binding, it's gonna bind with a CD4 accessory protein that's just gonna help with the binding that results in cytokine release and a communication between the two cells. And eventually that T cell is gonna clone itself and activate cytotoxic T cells or B cells. Okay, next is B cell activation. So B cells are activated when a helper T cell will bind bind to the B cell MHC and release cytokines that will activate the B cell. Oh, B cells will also bind to an antigen and present that antigen on its MHC molecules. So then the B cell will proliferate into plasma cells, which are effector cells and also memory cells. And the plasma cells will secrete antibodies that act right away. Okay, and antibodies, from the slide before, they have different functions. So one function is neutralization. So antibodies can actually stop the pathogen's functions by just binding onto it sometimes, or it can opsonize the pathogen by basically marking it for phagocytic cells and like facilitate their being taken up. And they can also participate in the complement system. So basically the antibodies will first bind to a foreign antigen on a cell, and then the complement system, which we talked about before, it's like a system of proteins that's gonna bind to the antibody, and then that's gonna embed itself into the cell membrane to form holes on the cell membrane. So there's gonna be a flow of water and ions into the cell that's gonna form a pore, and then the cell's gonna lyse and die. And antibodies can also mark cells that have to be killed by natural killer cells, which kill infected cells. Okay. Okay, so next is cytotoxic T cell activation and action. So cytotoxic T cell activation will require helper T cells and antigen presenting cells. So once they're activated, these T cells, so they're activated in a similar way. They will seek out infected body cells by binding to antigen fragments that are presented in the MHC molecules with the help of a CD8 protein. So these infected cells will also be presenting the same antigen through their MHC. Cytotoxic T cell is going to bind to it, and then it's going to release perforin, which perforates cell membranes with the holes, and also granzymes, which initiates apoptosis. And so that infected cell is going to die. Other things, vaccines, they will expose the body to antigens that are similar to that of a virus. That creates memory cells for a secondary immune response for the first time you're actually infected with the actual thing. Active immunity and passive immunity are different. So active immunity is when your antibodies are produced by your own body. And then passive immunity is when antibodies are produced by someone or something else. So an example of this is when the mother produces antibodies, it can get transferred to her baby through breast milk. And it can also come from animals like sheep and horses. They can produce anti-venom that will protect against snake venom. And antibodies can be used as tools. So monoclonal antibodies are antibodies that are produced by a single clone of B cell, and they can be useful for tests because there is a hormone during pregnancy that antibodies will bind to. Antibodies also have a role in MHC molecules have a role in immune rejection. So certain tissues, when you're taking tissue and like donating it to someone else, your tissue might not match up with that of the host. So when they have different MHC molecules, they can be recognized as foreign tissues and attacked by the immune system. And also blood type is very important because there are three different types of blood type carbohydrates, which are A, B, and O. There are 4 different blood types, which are A, B, and O. So these carbohydrates will create really intense reactions for individuals with other blood types because these carbohydrates match carbohydrates on other common pathogens that we respond adversely to. So we will also respond adversely to other blood types. Okay. This is just your immune system like not functioning properly. So allergies is basically, in short, I'm gonna go fast. Allergies is like a faulty response to things that shouldn't be that shouldn't cause an allergy response. Yeah, so that's gonna create anaphylactic shock, which is when there's a sudden drop in your blood pressure and constriction of airway, which is inflammation. That can be relieved by EpiPens and anti-allergy medication. There's also gonna be autoimmune conditions, which is when self-tolerance is lost. Oh, also self-tolerance is super important, which is when your immune system will not attack itself. It's gonna attack something else. And also immunodeficiency can result from AIDS. And AIDS basically makes your immune system like attack itself. And yeah, exercise and rest is really good for it. Okay, and here's just ways of invading the immune system. So the antigens on pathogens will change a lot, like in the flu virus. So the memory cells often aren't very effective. So for that, a lot of different vaccines will have to be made. Also viruses can be latent, so they just stop producing their proteins. They kind of become invisible to the immune system and they just don't go undetected. And then AIDS is caused by HIV, which reduces the T cell count until like you don't have an adaptive immunity anymore, which makes the human body susceptible to a lot of other different diseases that seem to be mild but can be really, really dangerous. And also there are cancers that can be caused by viruses such as HPV, and we get vaccines for those too. Okay, I'm good.